Hello, everybody. We are now on to chapter 17, which is blood. And blood, you probably have heard me say that this is the freeway system of the body. And it is. That's because your cells, all 60 trillion of them, it's a good question. How are we going to get nutrients to all 60 trillion cells? And the answer is, just like any other network, is that you have a shipping line. You have a way for carriers in order to get to their locations and drop off product. This is speed stack pumped, NO. Anything that says intense energy and 300 milligrams of caffeine is designed to get you going. So I've been drinking these since I was a teenager. Don't start buying them because the price will go up and I won't be able to find them. All right, so anyways, folks, yeah, as you can tell, it's been Sundays are my do a bunch of work day, so I'm doing a bunch of work, period. Okay, everyone, so let's talk more about blood. And it's interesting is that blood is, well, the movies at least, they show that blood, if you lose a lot of it, basically you die. That's exactly what happens because, as I said, your blood is the only way to get nutrients and essential items and also transport waste from all 60 trillion of your cells. And let's talk about the function of blood. And the first one, as you'll see on the outline, is transport. Blood has essential items that it must transport to cells and from cells. And one example is oxygen. Oxygen is designed for your cells to undergo cellular respiration, which is how they make energy. Also, your cells are very, when they produce waste, they do what everyone else does and throws it in front of their curb outside. That's exactly what cells do, is they put waste into the blood, and the blood must transport these wastes. Now, you're probably wondering what happens to these wastes. Well, gases just get exhaled, but other solids, they must be filtered by the kidneys. And also hormones. As you know, hormones are chemical messengers. They are chemical messengers. What their job is to tell cells to do things. For example, epinephrine, as you learned about in the autonomic nervous system, basically tells a bunch of your cells to coordinate behaviors, to change their behaviors. And this happens fast because your blood is pumped through your body at about 4 miles an hour under peak conditions. Now, next is regulation. And blood is actually designed to maintain internal body temperature. So maintain internal body temperature. So basically, your blood is shuttled throughout your body in order to make sure that essential organs are going to be properly, how should I put this, kept at a stable temperature. Depending on the environmental conditions, blood might shift from one part of the body to the next. Like for example, when it's really cold, blood will shift more into your core in order to properly insulate your internal organs. When it's really hot, it will move more into your integument, more into your skin in order to prevent overheating. Also, it acts as a buffer to maintain normal blood pH, and this is complicated. It's hard for me to say in just one sentence, but basically your blood has the ability to convert acids to bases and also to transport things and neutralize things that make your blood acidic. So it maintains normal blood pH. It's actually a very complicated process. Okay, we also have maintained adequate fluid volume. This is the only part right. Your kidneys really are the structures that make sure you have normal blood volume. But I guess you can say the blood plays a role in that too. Now your blood also has a number of protective elements. Protective elements. And what it does is that it's going to prevent blood loss when damaged. And blood is actually a connective tissue, believe it or not. And it's because it has a protein called fibrinogen that converts into an active protein called fibrin, and fibrin is responsible for clotting. It's like a long string-like structure. Whenever you have a blood vessel that's damaged, which happens pretty often, what's going to happen is that the fibrin is going to cause clotting. And what that's going to do is it's going to seal the defect, which will prevent further blood loss. Someone that's known as a hemophiliac does not have this ability, so they can have significant blood loss even from a small cut. And it also prevents infection because your blood actually has a number of protective elements too. So what it does is it has defensive proteins, such as one we'll talk about with the immune system, called complement. And it also has circulating leukocytes, which are white blood cells. Now, of course, if an infection propagates in your blood, it's called sepsis. Now, let's go ahead and talk about what the structure of blood looks like. And it actually looks like this. So we're going to say that this is a test tube. And actually, blood is maybe about, 45% of what we call hematocrit, which is mainly consisting of red blood cells. So this is about 45% hematocrit, which is mostly erythrocytes, also known as red blood cells. And it's also composed of things like platelets and similar structures like that. There's also something called the Buffy coat. It is a very thin coat, but it's mainly consisting of cells such as leukocytes and platelets. So that is actually correction, is platelets are not found in the hematocrit. To correct that, it's basically all erythrocytes or red blood cells. And the majority of your blood is actually something called plasma. And plasma is a white, kind of a yellowish, like the book says straw colored. I would have to agree with that. So plasma is basically consists of ions, nutrients, especially glucose, which is what makes it sticky, proteins, hormones. It transports a lot of things. Oh, and of course, gas. Well, actually, no. Gas transport is mainly the hematocrit. And there are some proteins that we'll talk about briefly. One is called albumin. And albumin is a rather large protein. It's mainly responsible for transport. It does transport some products in the body, but also it maintains what we call osmolarity. Because it is a very large molecule, what it does is it makes sure that when the heart contracts, the proper amount of fluid is going to stay in the blood vessels. So it maintains osmolarity. Something called colloid pressure, which we'll talk about later. Next to that is alpha and beta globulins. So alpha, beta globulins, and globulins are mainly mainly transport proteins. They also make up a protein called hemoglobin, which is the functional part of the red blood cells. So what they do is that they bind to things like, that's not how you spell binds. They bind to things such as lipids, nutrients such as vitamins and ions. Next, we have gamma proteins. And gamma proteins are immune proteins. We'll probably be talking more about these during the immune systems. And lastly, we have the protein fibrinogen, which, as we talked about, is going to be converted into fibrin, which is responsible for clotting. Okay, this is kind of a repeat of what we talked about earlier, but I do want to note that the blood is going to transport nitrogenous waste. Now, what is a nitrogenous waste? Well, cells are very, they produce a lot of waste products during their metabolic reactions. For example, urea, uric acid. These are produced from protein metabolism. Creatinine, which is a product of creatinine, it is a product of ATP hydrolysis. Ammonium is another waste product of protein metabolism. So this is going to go into the blood, and the blood is going to transport it to kidneys, where it will be filtered and excreted. Now, in the digestive tract, your body works very hard to convert proteins, fats, carbs, and nucleic acids into their basic elements. Into monomers or basic elements. Now, when they're converted into their basic elements, they're going to be absorbed into the blood. Actually, fats are mainly absorbed in the lymphatic system, but it will eventually get into the blood. So it is absorbed through the walls of the intestines, intestinal walls, and then it is going to be utilized by cells. Because, as I said, your cells are the things that need nutrients and energy. Also, respiratory gases. Respiratory gases. Mainly oxygen and carbon dioxide. So O2 goes to the cells, and CO2 goes away from the cells. Actually, that's confusing. So CO2 goes into the bloodstream. CO2 goes into the bloodstream. Now, CO2 is interesting because CO2 actually binds with hemoglobin, whereas whereas oxygen binds with the iron in the center of hemoglobin. We'll talk about that in a moment. And also hormones. Hormones are signaling molecules that are transported through the blood. So signal molecules. And these normally have effects on lots of different cells that travel through the blood. Travel through blood. Most signal molecules are rather localized, but hormones actually go through the blood. All right, let's go ahead and talk now about erythrocytes. Erythrocytes. And basically how these work is that they are very basic cells that actually are in the process of dying the moment that they're made. And they have what's called a biconcave shape. So they kind of are donut shaped. And the purpose of this is to maximize surface area. Cells ideally want lots of surface area with only enough volume. And this biconcave shape maximizes surface area. It also has no nucleus, and it has no, I'm going to abbreviate this, membrane-bound organelles. It's mainly filled with a protein called hemoglobin. In fact, it's packed with it. So it's actually 97% filled with hemoglobin. That is the functional unit of the red blood cell, also known as the erythrocyte. So hemoglobin looks like this. It's actually a combination of several different proteins. These are called tertiary structures in case you took bio before. So it's actually four hemoglobin proteins, and in the center is a molecule of iron. So these are the hemoglobin subunits. Hemoglobin subunits. And this here is going to be iron. Fe2+. And the iron transports oxygen. Now, there are a couple of different types of hemoglobin here. Go ahead and say that oxygen is going to be this nice purple molecule. When oxygen binds with hemoglobin, it's going to turn into a form called O2 bound. It's going to be called oxyhemoglobin, and it's going to look like, it's going to take on a more of a bright red shape. Blood is never blue. It's either bright red or sort of a darker red. When hemoglobin binds with oxygen, it is going to be a brighter shape. Now, when it is not bound with oxygen, it's still going to be red. It's just going to be a little bit of a darker red. So this is when it does not bind with oxygen, and we call it deoxyhemoglobin. And it's going to be more of a dark red shape. Now, when carbon dioxide binds with hemoglobin, let's do carbon dioxide in teal, it is going to be called carb, or carbon dioxide, amino, because it's binding to a protein, hemoglobin. I know that's kind of a long word. But this is the form that hemoglobin becomes when it binds with carbon dioxide. And keep in mind, it binds directly with the subunit. It does not bind to iron. Does not bind with iron. I think the problem with carbon monoxide is it does bind with iron, and actually the iron molecule prefers to bind with carbon monoxide. Our cells cannot use carbon monoxide for energy, So what happens is that slowly your brain begins to shut down because it isn't getting the proper amount of oxygen. That's why people who get carbon monoxide poisoning just go to sleep and don't wake up. That's why I suggest to everyone buy a cheap carbon monoxide detector. They're not that much; they're like $12 from Home Depot, but it could save your life, especially if you live in an area that has more, or not area, a house that is older. All right, now we're going to talk about white blood cells when we return. All right, everyone, for the second part of our lecture, we're going to talk about the process of hemostasis, which is what happens when you get, hopefully, just a minor wound. Or if something terrible happens, like a more major wound, this process will also save you from having excessive blood loss, hopefully. But it's my sincere hope that all of you only have things like paper cuts or minor scrapes. Or maybe your kitten scratched you a little bit too hard. Something like that. But anyways, what happens is that we have something called a blood vessel, which we'll be talking about very soon. And these blood vessels are extremely numerous in the body. And of course, they get damaged once in a while. Now, when they do, think of it as a hose getting a crack in it. Now, a hose is very similar to a blood vessel because it's trying to keep in something that's highly pressurized. And when that happens, usually what you think about doing is, okay, well, why don't I wrap something around the defect in the rope to keep in the water? Well, the events of hemostasis are rather similar. So we're going to write down the events of hemostasis. Events of hemostasis. Now, what's going to happen first is a stage called vascular spasm. Vascular spasm. Now, we haven't talked about this yet, but the walls of the blood vessels actually contain something called smooth muscle. Smooth muscle in the blood vessels does one of two things: constricts or dilates. Now, if you have a wound, so when a wound occurs, vasoconstriction minimizes blood loss. Minimizes blood loss. Now, this is important because the less blood flows through a damaged area, the less blood you will lose. So what happens here is that this is the diameter of a normal blood vessel. Let's go ahead and draw it in blue, no significance. We'll say that it has a radius of, I'll just say, 5 micrometers. I know that looks like some, but it's 5 micrometers. Now let's say after an injury, it could very well vasoconstrict to possibly 2 micrometers. And of course, when this happens, less blood flows through, meaning less blood can be lost. So what happens is that the smooth muscle actually decreases the radius of the blood vessel, minimizing blood loss. Now, what's going to happen now is that platelets, which are just small fragments of a larger cell called a megakaryocyte, what's going to happen is that you are going to have something called a platelet plug formation. So this is step 2, I guess you can call it. Here, I'll start to label these. Platelet plug formation. Now, platelets actually are capable of communicating with each other. So what's going to happen here is, let's say that this is the blood vessel, and this is a defect in the blood vessel. So this is the defect here. What's going to happen is that platelets are going to begin to collect here. What the platelets are going to do is they are going to send us a signal for aggregation. Now, when this happens, keep in mind that all the platelets are signaling this. So the more platelets that form, the stronger the signal gets. And the platelets keep on aggregating until the defect is filled. So this is an example of what we call a positive feedback cycle. Positive feedback cycles, what they do is that they keep on building in intensity until something stops it suddenly. Because if it keeps on growing and growing and growing, eventually it needs a stopping point, right? I just wrote positive feedback. Of course I did. Think of it as a snowball rolling down a hill. It's going to keep going faster and faster and faster and faster and getting bigger and bigger and bigger, and then it's going to stop because the hill is going to come to an end. What's going to happen is that these platelets are going to keep on coagulating, and then they're just going to stop. So when defect is filled, aggregation stops. So it keeps on growing in intensity, and then it just stops. Okay, and now we have something called step 3, which is called coagulation. Coagulation. See, that's written bad even for my standards. Well, I cannot spell today, can I? Coagulation. What's going to happen here is that floating in blood, there is a protein called fibrinogen. And you're going to hear this term a bit going forward, is anything that ends with OGEN means that it's inactive until it's active. So what's going to happen is that these platelets are going to send a signal that is going to take fibrinogen, which is inactive, and it's going to signal it to turn into long threads. These threads are going to reinforce the injury. So fibrinogen, which normally floats around in the blood and does nothing, is going to become active fibrin. And this fibrin forms threads that reinforce the platelet plug, as they call it. This is going to form something called a clot. All right, so to recap these steps, when the injury first happens, you're going to have vascular spasm. In order to minimize blood loss, the damaged vessel is going to constrict. Then what's going to happen is that platelets are going to begin to collect at the defect, and they're going to send signals to other platelets to collect at the site of the defect. As time goes on, more and more platelets are going to aggregate, called a positive feedback cycle. So first they're going to aggregate slowly, then faster, then faster, then faster and faster. And as soon as the defect is sealed, it stops. We call this platelet aggregation. Once the plug is formed, fibrinogen is going to convert to fibrin and form what's called a clot. Then, of course, after a while, healing will start. And that is the process of hemostasis.